Okay, I'm going to introduce Fairlie and we can get started. So thank you all for coming to the first GenPALS talk of this quarter. Obviously, we're not doing it in person like we did last quarter, so you're gonna have to make your own coffee. And today we have the pleasure of hearing from Fairlie Reese, who's a fourth year PhD candidate in the Department of Developmental and Cell Biology in Ali Mortazavi's lab. And her research primarily focuses on long read sequencing data analysis. So here today, she's going to tell us about a software package called Swan that she developed for doing downstream analysis of long read sequencing, especially to analyze RNA splicing. So take it away, Fairlie.
Thanks. Well, I probably don't even, I can't share my screen. I made this Zoom link.
I have to make you a host or something, right?
You just have to, okay, you press the little green arrow by the share screen button, and then you hit multiple participants can share. It's
green arrow by what?
Okay, here, let me just come and do it.
Dude,
for you.
I'm such a boomer. I'm sorry. Okay, one sec. Sorry, y'all.
No, you should be able to do it, Fairlie. I can do it on my end.
No, I am logged in.
It?
I think I'm logged in on like my normal Gmail account instead of my UCI one. So that's the problem. Okay.
Is it working out? Oh,
can y'all see this?
Yes, we can.
All right. So
it's like,
thank you,
I don't
Sam.
know if it matters, but I see like the Google Chrome top thing.
I don't know how to get rid of it.
It's fine. It's really small.
Let's not worry about it.
Well, the way to get rid of it is
I'm not going to do that. I don't need to have another application running at all times.
I'm just saying.
Okay. So yeah, thank you, Sam, for that great introduction. I feel like I don't even need to give a talk anymore. That was just pretty much all that I've done. But yeah, as Sam mentioned, today I'm going to be talking about my software package called Swan, which is a Python library for the visualization and analysis of long-read transcriptomes. And there are kind of like two parts to this talk. The first of which is actually just going over what Swan does and what it's used for. But the second part, which is kind of something that I've been asked to do a few times, which is just going over kind of like the lessons that I learned and advice for how to write a software package. So if you're not going to write a software package, maybe you can tune out for that part, but it might be useful for some people here. Okay, so first I'm just going to go over what sort of data we use with Swan. And so Swan is designed for using long-read RNA sequencing data. And long-read RNA sequencing data is advantageous compared to short-read RNA sequencing data because we can capture full-length isoforms. So for instance here, where we have, can I click on my presenter view? No, I can't. Great. So where we have like this gene up top that contains these two different isoforms, one which contains this red and green exon together, and one which contains neither of the two. In short-read RNA sequencing, we can pick up reads that sequence across these exon-exon junctions so that we can tell which exons occur right next to each other in the same transcript isoform. However, when splicing gets more complex, such as the kind of like a mutual inclusivity of these two exons, we always see red and green together. Short-read RNA sequencing falls short because we can never tell in any one specific read that the red always co-occurs with the green exon in the same isoform. And so long-read RNA sequencing circumvents this issue by simply sequencing through the entire length of the transcript molecule, giving you the unambiguously resolved full-length structure of each transcript isoform. So long-read RNA sequencing is not as new as it was when I started my PhD, but at the time it kind of was. And as such, my lab that was interested in generating and analyzing this sort of data was also interested or kind of had to or forced to develop tools that are specific for analyzing long-read RNA sequencing. So my predecessor in the lab, Dana, developed a tool kind of for the first step of processing of long-read RNA sequencing data, which is called Talon. And basically what it does is it takes each read in your long-read RNA sequencing data, and it tells you which isoform it came from in your annotation. And an important part of pretty much any long-read RNA sequencing software to be useful is to report known transcripts, so transcripts that are present in the annotation, but also report novel transcripts or transcripts that are not annotated, not known before, especially because our current genome annotation or transcriptome annotations tend to be very heavily based on the previous generation of sequencing or short read sequencing. So basically we expect to find novel things in the long read sequencing data that are not in the short read sequencing data. So that is what Talon is used for. And so kind of like on the heels of that, or I guess kind of after that, we were like, okay, we have this way to process our data, but what are we going to do now that we have it processed? Now, what are we going to do now that we know what these transcripts are and what genes they come from? And that's where I kind of came in with this project, was developing a tool to kind of do downstream analyses of long read transcriptomes. And so as a broad overview of what Swan does, it uses one of these like weird hairballs you see down here to represent transcriptomes as graphs. And the sort of analysis tasks it can perform are things like differential gene and transcript expression, differential isoform expression tests, or as I will more commonly refer to it, isoform switching tests. It can find novel exon skipping and intron retention events. And finally, it can provide some really beautiful and interpretable visualizations and reports, in my opinion, although I am biased. What you probably don't know is, or what probably doesn't matter to you, is the distinction between like the new and the old Swan features. So in recent years, last year, I made Swan work a lot faster, and it has a much easier workflow for initialization of highly complex data sets. And in addition to tracking isoform abundance, it also now tracks transcription start site and transcription end site abundance. So how often is one 5' end used versus, or how often is one 3' end usage. I also implemented a more robust statistical test to find these isoform switches, as well as find transcription start site and transcription end site switches, using a framework that was not developed by myself, but still worked very well in our hands nonetheless. I upgraded it to use, instead of a pandas data frame, the ScanPy data format called ANData to store complex metadata along with many other things, which in turn kind of makes Swan compatible with both single cell and bulk data, which was a pretty big hurdle that I wanted to clear given the advent of our lab starting to generate long read single cell data. And in addition, I have implemented new plotting options. So I'm going to go over a brief overview of how some of the things in Swan work, what they look like, and then kind of show you a brief set of applications of Swan to a publicly available single cell long read RNA sequencing data set that my lab has generated. So what does that hairball that I showed you a couple of slides ago actually mean? And so I'm just going to kind of take you through how these graphs are constructed. So if you have a transcriptome annotation, which I have represented here, these two transcripts that belong to the same gene in this like dark turquoise color, and you can see that one of them contains this exon and one of them contains this exon skipping event. Basically, our job is to transfer what these things look like in their traditional browser format to this, kind of hairball down here. And so the first thing that Swan does is it finds each unique splice site or transcription start or end site, and it creates one of these little circular nodes that you can see down here in the Swan representation of the transcriptome. And these nodes, for all purposes, are colored essentially by their role in the gene. So the blue nodes represent transcription start sites or 5' ends. The orange nodes represent transcription end sites or 3' ends, where the yellow nodes represent splice sites that are internal to the transcript model. So once we have the nodes, we then need to represent whether or not introns or exons kind of connect those two nodes. And so we can do this by finding our unique exons and introns in our transcriptome annotation here, colored by these green and pink lines, and therefore kind of translated to the Swan representation here as well. And so this, you may be like, why the hell am I even like bothering to kind of reinvent the wheel here? And basically the thought behind this was not necessarily as a replacement for like a genome browser style representations of transcripts, but kind of like a supplement for it that more, it really empowers users to be able to visualize smaller differences in splicing than what you can see on the genome browser, or using like these, box and stick models of transcripts. So in the case where you might have like a really small indel at the end of an exon, and that's the only difference between one transcript and another, you might not be able to distinguish them in the traditional genome browser style format, because the difference might only be, a few pixels on your screen. However, in the Swan representation, that small indel would translate to the addition of an entire node. So you can tell the difference between any two transcripts by really just like examining which nodes are included in them versus not. And so that's kind of the power of this plotting style. Okay, so what does this look like?
Fairlie? Sorry, can I ask a question?
Mm-hmm.
So you have assigned each unique splice site to a node. So basically a node is like the beginning and or end of either an intron or an exon, right?
Mm-hmm.
How do you know that? Is that just something that we know from a reference, or does your software actually figure that out from scratch?
So it's basically as an output from Talon, you get a GTF transcriptome of all the transcripts that are seen in your data. And that is a combination of taking what is in the reference, right? As well as identifying what is new compared to the reference, and kind of coming up with these novel transcript structures based on where and where like the splicing occurs within the reads as well. So I would say the majority of these nodes are going to be from the annotation, because the majority of splice sites we very, very well know from short read data. But that's not to say that all of them are. I don't know if that answers your question.
Yes, it does. Thank you.
Okay, great. All right. So this is kind of a simple toy example of what these look like, but what do these look like in practice? So here is an example of these two kind of like basic units of graphs that Swan creates. The first on top here is a gene summary plot for the gene CSF2AR. And what you're seeing here is essentially a high level view of all of the different splicing events that happen in the transcripts that belong to this specific gene. And here, what I can point out to you is that you can see that there's a lot of different alternative 5' ends. So each one of the blue nodes here represents a start of a transcript that was seen in the data or in the annotation. And all of these orange nodes here represent an observed or annotated 3' end seen from the data. And here I've also chosen one of the Swan plotting options is to highlight what is novel compared to the annotation. And so the dashed introns and exons here indicate introns or exons that are not seen in the annotation. And the outlined nodes here indicate splice sites or TSSs or TESs that are not seen in the annotation. So there's not too much novelty in this gene, but there is this basically novel splice site seen here, which in turn kind of propagates to this novel exon, intron, and exon here. So instead of looking at a high level summary of your plot, you can also look at the path that a specific transcript has through your plot. And so this here is essentially the path between introns and exons that one specific transcript belonging to the CSF2AR gene takes through this graph. So it will always go exon, intron, et cetera. Okay. So now I'm going to go through a brief rundown of the data that I'm going to be showing in the upcoming slides. So my lab's first attempt at generating single cell long read data was applied to the C2C12 cell line model of myogenesis, or essentially muscle maturation/formation. And the details of this differentiation process aren't really important for your purposes, but essentially what I want you to know is that we have these undifferentiated myoblasts, which will be shown in pink for the ongoing part of the rest of the presentation, as well as these two different subtypes of these differentiated cells that we found, which we called either myotubes or mononucleated cells. The former being kind of what we canonically think of as the more mature version of these differentiating muscle stem cells, and the latter being the kind of in-between state between the myoblasts and the myotubes. So we plot these just briefly because it's single cell data, we've got to show UMAP. We essentially found these different clusters on the gene level, and we used these clusters to inform our cell type labeling of those aforementioned cell types that I mentioned on the previous slide that I'll be using to basically perform the analyses between in the coming slides. Before I do that, I want to circle back to this idea of isoform switching. And this is because it's kind of like an esoteric idea for people who have been kind of living in differential expression land for the majority of their lives, or maybe even don't even know about these sort of things at all. So basically, isoform switching, in comparison to like a gene expression test, it's not just looking for, whether it's there in one condition and whether it's not, or like down in another condition. It's looking specifically for genes where the major contribution to the expression of that gene changes from one isoform to another isoform across these conditions. And so here's a really nice example that I'm showing in the bottom left of this slide over here, where this isoform switching gene over time, essentially the majority of the expression of the gene can be attributable to this green isoform at this earlier time point. Whereas as time moves on, it switches such that the predominant isoform contributing to that gene's expression is now the pink isoform over time. And so it's a little bit more of a nuanced idea or compared to differential expression, mainly because we don't necessarily want to exclude differentially expressed genes from being analyzed, but more so that it's based on how much of the isoform's expression is attributable to each isoform each gene's expression is attributable by each isoform that makes up that gene. And as a brief rundown of what the statistical framework looks like, which again, I did not develop, this was developed by a peer in a different institution. It does a chi-squared test of proportions on the expression of isoforms that are within the same gene. And instead of like log fold change, which is kind of the comparable metric that you think about for differential expression testing, you calculate the change in proportion isoform usage across the conditions that you're comparing. And then again, similarly to differential gene expression, you can threshold these genes based on their p-value and the change in the proportion isoform usage across your conditions. Okay, so using our different cell types we identified here, so myoblasts, mononucleated cells, and myotubes, I performed isoform switching testing, TSS switching testing, and TES switching testing between each of these different pairwise comparisons. And here's just an example of how many isoform switching genes I found for each of these different comparisons. There aren't very many. This is the first data set that we generated that was long read single cell, and it's honestly not that great. So I think that probably is a large driver of these low numbers here, is just that we didn't have enough to call things as statistically significant. But I'm just going to show you some examples of what did come from these categories for the purposes of this. Okay, so this is just for isoform switching, but the things that I'm going to highlight are from TSS switching genes. So this is a gene report that is from Swan. Essentially what it does is it plots the transcript model alongside you can either do expression or percent isoform use. I typically find that percent isoform use is the more informative metric to look at if you're trying to like see how the isoform switches or TSS switches are working across the different conditions that you've identified. And so here, what you're seeing is that basically the myoblasts, which are shown by this little pink category here, broadly tend to express this like longer isoform that uses this 5' end or something in the realm of where this 5' end is. Whereas when you start to differentiate, you get into the MNCs or the myotubes, you can see that the majority of the isoform usage is really attributable to one of these shorter isoforms here. So that's pretty interesting. And yeah, this is a plot that just comes right out of Swan that there's a lot of different tunable parameters. And so this is, I don't know, I think it's really cool and pretty to look at. And I think it's publication ready, but maybe other people would think differently. And just one more example of what one of these reports look like. This is actually the gene that we chose to highlight in the paper that this data comes from. This is another transcription start site switching gene called TNNT2. And similarly to the Spark gene, it basically is showing that there's a high preference in the myoblasts to express these really short isoforms. So this one here, which is like that one's ridiculously short, as well as these two here. Whereas in the mononucleated cells in the myotubes, there's a higher preference to express these longer isoforms with these upstream 5' ends. So pretty cool that we can identify these robustly with Swan right out of the box. Okay, just a few more slides of data. So I also just want to plug the fact that since I used the AND data format that is kind of underlies ScanPy, you can just put what Swan, how Swan represents things and like put it into ScanPy's plotting functions and it just works, which is awesome because that means that I didn't like have to code my own like matrix plots or dot plots or UMAP plots or whatever. So this is actually from a different data set, but this is just showing how you can look at the expression of all of the different isoforms that belong to a different gene across conditions, the different TSSs that belong to different genes across conditions, or TESs. And of course, this strategy also enables you to be able to make these like traditional single cell style plots, which I'm sure Emmanuel is going to have a fit about this violin plot, but it's more of a proof of concept. Single cell long read data particularly is extremely sparse, but you can make violin plots. You can color your UMAPs based on expression of specific isoforms or of specific transcription start sites or transcription end sites. So this is going to end the first part of my talk. So if anybody has any questions about like kind of what Swan's capable of, or like how it works, or what the hell did I even just say, maybe we can break for that for a second before I move into the next part.
Can you use Swan on short read data?
Yes. Okay. So you can use Swan on short read data. So obviously, Swan was motivated for the use with long read data, but if you can generate a GTF transcriptome as well as a transcript level abundance matrix or counts matrix of your short read data, you can use it with Swan. And it's actually something I'm like trying to collaborate with because it's not as simple usually to get those things out of short read data. Typically people I think use Callisto to get transcript level counts, but I'm not sure about GTFs. But yeah, so it has been done and it probably will continue to be done, at least by me. Yeah, if you're interested in studying splicing your short read data, you might consider this, or at least making nice plots. Okie dokie.
Can I ask a technical question? So I understand, I think, how you did the stats and the relative abundance stuff, the proportions for bulk. do you still use the same idea when you're doing single cell? Is it just like you're just looking at cluster to cluster?
Yes. Yeah, I know what you're saying. So this strategy, the strategy that I actually mentioned was developed for single cell, but
Okay.
presumably due to the sparsity of the data, it always works by basically aggregating first. So in the case of like my data, these are like the values that the statistical test would be working on, because I was always comparing like these different cell types. So essentially you need to define groups on which you want to like make the comparisons first. And essentially what it'll do is it'll just aggregate everything from that group to calculate these percent isoform use values before performing the test.
Okay.
Yeah.
Does it deal with zeros at all? Do you know?
It doesn't do anything with zeros.
Cool. Thank you.
Oh boy, that was dangerous. Any further questions? If not, I'll move on. Oh yeah, for sure.
Very basic question.
Mhm.
So is this like a package? Like do we treat it like if someone wants to use it, how do they go about using it? Do I like, you know how I install packages in R? Is it like that?
Yeah, so Swan is written in Python, and I'll go over this a little bit in the upcoming slides, but basically it's instead of through like bioconductor, which is for R, of course, I have it hosted on both GitHub and on the Python package index, which is called pip. So you can pip install this package, or you can download, or you can clone it from GitHub and you can pip install it from the directory itself. But one of my goals was to make this very, very easy to install, and I think I achieved that. So yeah.
Awesome. Thank you.
Mm-hmm. Thank you, Nargis. Okay, so on the heels of that, I'm going to kind of go over what the process and lessons that I learned from writing software in grad school were. And so these are kind of like the key points that I distilled down from like a lot of work, which I'm going to go over one by one and go over like some greatest pieces of advice for these things. So the first of which being design principles, then installation, documentation, testing, publication, and maintenance. So let's just go ahead and get right into this. In terms of design principles, I kind of really only have one example to talk about because I'm not going to talk about how all of the different class and function structure is set up in my package. But essentially the biggest thing here is that I recommend that if you're writing one of these, you should take note of what others in the field are doing or what sort of structures people are using. So in the first iteration of Swan, I used typical like data frames to store expression, etc., whatever. And this didn't really enable me to do really complex tasks such as storing metadata beyond just like a single sample name. So obviously in like more complex data sets, you can have lots of different biological variables that you might want to base different comparisons that you perform on, such as like sex, condition, time point, et cetera, et cetera. And the strategy that I used initially didn't really enable that at all easily. And so when I set about kind of reworking Swan last year, I was coming off of doing a lot of single cell analyses in Python, and I thought that it would be a good idea to just adapt this and data structure, of which the details that you see over here are not quite important, but I kind of like this visualization. And so essentially what this enables me to do is instead of like having to have made my own class from scratch, I basically, I still made my own class from scratch, but I store a lot of what's in there in one of these, such as expression, metadata, proportion isoform usage, and statistical test results as well. And not only does this allow me to like do kind of what I was really setting out to do, which was storing multiple levels of metadata, especially for like single cell purposes, where you have a cell that might have like a cluster, an experiment, a sample, a batch, like whatever, but also allows me to like seamlessly integrate with ScanPy plotting functions, and allow me to use Swan not only as a bulk tool, but a single cell level tool as well. So that's kind of, beside, if you want to talk to me about like how I structured actual classes and stuff like that, I probably don't have much intelligent to say about it, but I'd be happy to hash it out with you. I just didn't really know where to start on design principles pertaining to that other than like this. So I'll go ahead and leave it at that. Moving on to installation, which is what Jesse was talking about, or what Jesse brought up earlier. Make sure your library is easy to install using a package manager. And as far as I'm aware, the options for this in Python are making your package installable either through Conda or through PyPy, also known as pip. And for R, it's making your package available to install through Conda or Bioconductor. So for me, I uploaded Swan to pip, and I also basically have a suite of files on my GitHub repository for Swan, which allows you to clone the repository and use pip install from that directory itself, instead of like having to pull something down from the web, which makes it really powerful because I'm able to let my users pull the latest commits to install the latest commits to my repository without having to actually go and upload a new release to pip, which is a fair amount of work, as hopefully Narcus is finding out today. I know she's working on getting her package published or on pip as well. Maybe I should have linked Swan here so that you can see the installation instructions. I'll probably go in and do that after the fact so that you can see what that actually looks like in practice. But if anybody else is interested in trying to upload their software to pip, these notes are probably okay. I wrote them like years ago at this point, because I was never able to find a comprehensive article with step-by-step instructions, and I was just like, I just need to write this down somewhere. So if anybody wants to, they can click on that. Okay. Moving on, my favorite part of this talk is documentation. You can never document too much. Swan has documentation on many different levels. I think what's really important is to basically make sure that every individual like block of code or like function is well documented, but also include high-level documentation that enables your users who may not be able to like understand the nuances of what like calling a function means to succeed as well. And so I'm just going to kind of go through the levels of documentation that I have for Swan here. So the first of which is function level doc strings in the code. So this is my actual code, and for every function I have these like these lines between these three double quotes, which tells you exactly what the input is and exactly what the output is of the function. And something I've recently realized, because I didn't, you should write these things as you go. It's a lot easier to write the documentation for a function if you're like using it right then and there. Even if you have to go back and revise it later, it's way easier than having to write all of your documentation from scratch and be able to like isolate the function, figure out what the output is, et cetera, et cetera. So again, write these as you go, in my opinion. I also used a website essentially to host the rest of what the documentation I'm going to be going over is. I specifically used Gitbook as the tool to host this website. It allows me to basically edit Markdown and YAML config files in my GitHub repository for Swan itself in order to create a website structure and pages. So I recommend that, although I know that there are a lot of different options for it. I also have API documentation hosted on my website. So this is essentially what you're seeing in the doc strings, but on an HTML page instead. And I basically used this really cool tool. I think it was this one, maybe it wasn't, to make these, where basically it parses your entire package and it creates a markdown file with the doc strings that you have already written, right? Because you were totally writing them as you went. And basically I was able to embed these in the website edit pages. So that's kind of that's the low level stuff. What about the high level stuff? Well, I have tutorials written that I wrote in Python notebooks, or like Jupyter notebooks, that are these combinations of markdown, text, blocks of code, and images, such that you can like help your users, you can help guide your users through like a series of tasks that they might want to perform while swapping their data in. And so I think that this is really useful, basically, to be able to like read, this is what we're doing, this is why we're doing it, this is how exactly you do it, this is what the output looks like. So I think that that's really important. I also included like an FAQ section on essentially how to read the hairball, because I think that, again, it's kind of intimidating to look at. And I have a lot of experience looking at these plots and I know what's going on in them, but it's easier if somebody teaches you how to read them than trying to figure it out from scratch and like drawing them out yourself, which is of course what I've had to do in the past. So I have this whole section on how to understand these visualizations. And then last but certainly not least, please include file format specifications in your code. I cannot tell you how many times I've tried to run bioinformatics software and it just hasn't worked because you told me that for this input argument, I just need to put a TSV. I need to know what the column names are. I need to know what the values need to be, et cetera, et cetera. So Swan, I think, uses like 5 distinct different types of input files, and I have them all meticulously documented here so that nobody can mess up, hopefully. Yeah, okay. And you can go, I mean, I'll put this link in the GenPALS chat afterwards, so you can go ahead and look through this as you wish. Okay, in terms of testing, more people should probably test, including myself. PyTest is a testing suite that allows you to essentially like test how your functions are generating output and whether or not that's the correct output. My current tests that I have implemented are not really comprehensive, but I have essentially done like, I've tested essentially the lowest level code to make sure that I can be confident in how Swan is calculating numbers, how it's doing its statistical tests, and how it's constructing the underlying data structures. But it's, again, by no means comprehensive. And to a certain extent, I don't think it would be worth my time to figure out how to like test making plots, because that is notoriously difficult. And furthermore, your PI probably doesn't want you to test your code because that's valuable time that you could be spending doing some other things. So this is kind of the compromise that I ended up going with. And I do get issues on my GitHub and I have to fix them, but I can be confident essentially in like the lowest level stuff on my repo. And just another note, I don't know if this is going to factor in. You can't test private functions with PyTest, and this is apparently considered bad practice. And I've just avoided this by not using private functions. Maybe that means that I'm writing my functions incorrectly. I don't know.
Fairlie, what is a private function?
So a private function is a function that you should only be able to call within like the code that like you, the developer, write in the library. So basically a user that downloads your software will not be able to call it. So like things that you're worried about, like the user like running and breaking, you know, some representation of their data that they have.
Okay, interesting.
Yeah.
There's always a simple example of that. Imagine that you have like the bank account, you have a class that is bank, and then you store your like balance for each account. So you don't want to be able to, like each person can change the balance of their account. So you define the set balance as a private function. So no one owner out of the class can change the balance in the class. This is somehow like the private function or private value or variable.
Mm-hmm. So like in the instance of my code, I have like a lot of helper functions that I have that are called by like parent functions, which are the functions that the users should be able to call. The users should never have to call these like child functions at all. And if they do, things will break. But for the purposes of being able to test them, I wasn't able to make them private. So instead, they're just not documented and not in my API documentation, which I had to do manually. So I would probably do this different in the future, but it would have required me being able to like test the, like make the most high level functions testable, which then kind of like negates my efforts of trying to test, you know, not necessarily the bare minimum, but like not everything, if that makes any sense. Anyway, it's probably not something that a lot of people have to worry about. Okay, last couple of points. Publication. Definitely consider what sort of paper your software deserves. This is something that I'm continually salty about, and I've definitely given my lab mates advice on this. Swan is a 2-page paper where part of it is like the references. Part of the second page is the references. And I've spent like hours and hours and hours writing this software and improving it. And it is so much more than what is in this one manuscript now. And it's just kind of a bummer that this is the way that it exists in its published form. So if you can, I would advise waiting for your software maybe to mature for a while before publishing. In which case, actually, Sam was the one that suggested I put this point on here. If your software is not published, but you want people to be able to use it and cite it, you can use a tool or a web tool such as Zenodo, which will generate a DOI for a GitHub repository release that you have. You can make that citable essentially before you even have a publication. And then furthermore, it's worth considering what sort of article you want to present your software in. So a lot of people throw their software in kind of as like a side of like a big paper. So they're like, okay, we did like all these analyses and then look, we like developed this new one, and it's like, it makes up like a paragraph in the results or something like that. But it also, you know, you might want to have it be its own standalone paper as well. So definitely make sure you take the time to consider this. And so kind of intertwined with this is maintenance. Just because it's published doesn't mean you're done. So basically, I've kind of alluded to this several times before, but Swan in 2021, in 2020, only could like make these crappy little plots, the one that's shown here. I mean, it's not that crappy, but and that's what's published. But I spent like a good month and a half last year fixing it up and making it relevant again for the complexity of the data that we have in my lab. And so this is kind of like the glow up that you're seeing here. Like, look at all the crappy things that Swan could make only in 2020. And now look, it can work on single cell data. You can plot the metadata as colors. You can change the colors of this for whatever reason. You can change the color of this. You can add the numbers. You can use the isoform names. You can plot percent isoform instead of log2 TPM. There's so much more that it can do. And I think that this is kind of a testament to the fact that if you want your software to continue to be used, at least by your own lab, because let's be honest, nobody's using it except for my lab, you need to be willing to be put into the work to make it relevant. So in the case for me, it was making Swan really work with single cell data, because there was no way it was going to work with single cell data before I went and rehauled. And I didn't even talk about this, but yeah, obviously make sure you address bugs that people put in your GitHub issues, and you can create new fix releases with these bug fixes. Okay. With that, I'd like to thank my predecessor slash like software developer, like person who gave me advice in my lab, as well as people that generated the data, as well as other people in my lab. I also wanted to give a special shout out to my friend from the Tilgner lab at Weill Cornell in New York, who helped me implement her statistical framework. But also, and this is very tongue-in-cheek, but I just want to thank myself. I am the only contributor for the software repo. This is just a graph of my contributions for it over time. So yeah, I just want to, give myself credit where credit's due. And if anybody has questions, I'd be happy to take them.
Could you?
That was
I
excellent,
want to hear.
Fairlie.
I want to hear your comments about like why, okay, like you chose to make Swan its own paper, right? But the other options, like you were saying, is like you could just add it on to a different paper. What do you think the like pros and cons of each of those things are?
So I think the big pro is pure numbers, right? So if you have a paper that is just Swan, and then you have a paper that's like, we use Swan to do X, that's two papers. That's more than one, right? I also think that it's probably beneficial in terms of like, if you're talking about like authorship, you'll probably be first author on a paper that is your software that you're writing. Whether or not you will be first author on a paper that just uses your software, even if it was developed for the specific use of that paper, that's not necessarily the case, you know? I do think that it's unfortunate that I wasn't able to use a more interesting biological system to profile when I wrote the Swan manuscript. But of course, I'm getting the chance to do that in my lab right now. So I don't know. I think that I would prefer having a standalone paper personally. I just wish that it hadn't happened so quickly, at the risk of sounding a little bit, you know, ungrateful that, you know, I have a first author publication that came out in my second year. Yeah.
Do you also have any comments about the different like types of paper? Like yours was the application note, so it's a really short 2-page, but you could also just have a full article about the software, right?
Yeah.
Do you have any ideas about like what is the difference? Like what does it take to have a bigger paper? Well, maybe what would you have had to do? I don't know.
Yeah. I mean, I'm going to be honest, I struggled to get this to fit into 2 pages. And I think that if I had had Swan in its current status and been able to write a paper, I would have been able to make something that was so much longer, particularly for the like isoform switching implementation alone, being able to kind of show the workflow of it, not just like one figure that's like kind of the apex of all of the analyses that Swan was capable of doing at the time. And to be honest, since I haven't written a paper like that, I don't really have a great grasp of what sort of like stories that they tell using software, but that's just probably how I'd go about it, is be more detailed. I'd probably I'd probably do something more along the lines of what I showed for this presentation, like, oh, here's the Swan graph, here's the transcript path graph, here's like a report for the TPM, here's a report for the percent isoform usage, and then like show some plots that maybe follow up using the numbers that Swan reports, but not necessarily plots that Swan can make out of the box, such as, like this one. I really like this one. I think that this is a very clear way of visualizing isoform switching.
To that point, Fairlie, have you thought about just publishing like a Swan 2 paper with all these changes and then publishing something?
I have been shot down by my boss about that idea.
You could just do it on your own.
I could, but I do not have time. Maybe later.
Like just as peer review, basically based on your GitHub repo. Like it's very minimal writing effort from what I've seen. Like, yeah, there's a software I use where they've gotten 3 papers out of it by just doing a new version like every few years or something. And the latest was like 2020. Yeah.
Yeah. If you want to share some examples of that by sending those papers to me, I'd be very interested. Maybe I can go renegade or go rogue on my lab.
I mean, even closer to home, like DESeq and DESeq2, right? Those are 2 separate papers. Like, yeah, it wouldn't be unprecedented.
Yeah, it's definitely not.
Yeah.
Probably too expensive.
It just requires more than
publication fee.
Yeah,
on your own, right? Without the lab.
And I had a friend whose goal was to do a single author paper, so I think for some it's a very desirable situation. But yeah, this was lovely, Fairlie.
Thank you.
Fairlie, can I ask you kind of a detailed question? So what I found about a lot of packages is that they write documentation as if you already had background, right? So like if you look at PyTorch, if you look at SciPy, like all of these things assume that you already know what's going on. And so when you were writing your documentation, did you ever think about like who is going to use this? Like, did you write it for like people who didn't know anything? Did you write it for experts? Did you write it for somewhere in between? Like, how did you think about that?
I think that's a good question. And the answer is that I think I tried to carve out niches in the documentation for both types of people. So I will just start by saying that the examples that you gave of SciPy and PyTorch are typically more like low level, people that aren't bioinformaticians are not necessarily using those with a lot of finesse on a daily, or sorry, people who are bioinformaticians are not using those with a lot of finesse on a daily basis. Like obviously people who have a lot more familiarity with like the low level details of how Python works or how just general object-oriented programming works might be able to parse through like API documentation with relative ease. And so for that reason, I have API documentation available on my website. But I also have like these comprehensive tutorials which not only outline like how you're supposed to go from like point A to point B in a set of like analysis steps that you'd want to run, but also like how to interpret the visualizations that are output from it. And maybe it's even worth like looking at my website for this. So similarly to how you might see on like the ScanPy package documentation, I have my like API documentation here. This is like the nitty gritty, like what arguments go in, what comes out, like whatever. This is probably more similar to what you'd expect to see for like SciPy or PyTorch, like you mentioned. But I also have like these tutorials, which kind of like they go through like, oh, this is how you download data. Like this is how you prepare the data to be, run with Swan. And then, what happens if you want to perform your isoform switching test? Like, here's the details of how it works. Look at your data, set it up, do the test, look at your output, filter your output, et cetera, et cetera. So I really tried to make there room for both experienced and novice, I guess not necessarily novice, but like, novice programmers to use the software, because I think that as genomics people, we kind of occupy this unique intersection between those two disciplines. And I think that as somebody who has a strong handle working with the people that actually employ these software a lot, I know what sort of documentation people are looking for and what's the most useful for these sort of people. Maybe I don't, maybe you think my tutorials are terrible, but I tried to make them not terrible.
Yeah, cool. Thank you.
I have a question. I was just wondering what kind of single cell short read data you've applied this to, and if it's working as you would expect.
Yeah, so I have only applied it to bulk short read data so far, because it's also supposed to work with bulk and single cell. I've single cell data on a couple of SAM's projects, particularly that are interested in studying splicing. One thing that I would love to update in the future, though, is that it does not use sparse matrices, and that is kind of a really big obstacle to overcome when you're looking at more than the number of cells that I have, which is 464 cells here. There are ways that you can get around it by basically like aggregating your cell types a priori before adding them to Swan. But if I had another like 2 months to dev this, I might try to make it work with sparse matrices, but I just don't see that happening anytime in the future.
Is the Wait a minute. Can you unpack that? Is long read single cell inherently not sparse?
No. It is sparse. I just don't work
Okay.
with sparse matrix. Like I literally, the data type sparse matrix is not what underlies the representation here. It's a dense matrix.
I see. Okay.
Yeah.
Doesn't that
So therefore it
like,
takes up more memory.
you're using and data, right?
Mhm.
So can't you just cast the type to a sparse matrix? Like, is it not that easy?
So because I'm a noob, a lot of the calculations that I do on it, I use the dense matrix representations of in pandas because I'm able to use like group bys and so on and so forth, especially for the percent isoform usage calculations, which are not super simple because you need to group your observations by what gene they came from, as well as what cell type they came from. And I haven't done too much thought into how it would be to work with sparse matrices, but working with NumPy arrays kind of scares me. I like having labels. And so that's what has prevented me from doing such.
It seems like
But
the
you
and
don't
data
like
is nice.
design matrices?
What was that?
You don't like design matrices?
I have no idea what that is.
It's just like a matrix in ML. Go ahead, Sam.
I think like and data has the obs table with all of the the group labels and stuff. So I don't know what ScanPy is doing, but they've somehow figured out a way to
I have
work
no
with
idea.
both sparse
They
matrices
figured
and
it out,
have
but they
pandas
don't
work.
right, but they don't have they don't have percent isoform calculation. I could definitely implement like the TPM normalization in ScanPy, because that's just like using the God, what is it? I don't even normalize total, or is that SROT? I don't know. Function, but
have that,
the pi
yeah.
or the percent isoform calculation is a little bit unique, and I'm not sure how many people are using it at this point. And it could be different based on what different piece of metadata you're using, you know, because you're calculating it usually not on the level of single cell, but on like some pseudo bulk level, like cell type or cluster.
Yeah,
So
I
I'll
don't
just say
think.
that. All right, go ahead, Emmanuel.
Good.
Sorry.
Good. No, I was
I was
just
gonna
gonna
say
say
the
that
hour's up.
it's not. Sorry,
Should I
it's
mute
getting
one
late
of
in the
you?
day, and I think we're all getting tired. Go ahead, Sam.
No, you need to go ahead first because I was gonna let people leave because it's already 4, and I want to keep people longer than an hour.
Okay. Well, you can do that then.
But you had a comment. Okay. I will just say before we like disband that we're still looking for some. If you want to speak like at the GenPALS seminar during either the rest of the winter quarter or sometime in the spring quarter, you are welcome to get in contact with us. If you want to talk about any bioinformatics thing that you're working on, we would love to hear it.
Yeah, we're tired of talking. We want you guys to talk too. Okay, Emmanuel, want to give your comment now?
Yeah, I mean, it's really uninteresting, but I was just going to point out that I'm currently working with like and data objects and like plugging it into like different things. And it's like it's not always immediately obvious whether or not the functions you're plugging your sparse matrices into can think about sparse matrices.
Yeah.
So I think you're right in saying that it may not work out of the box. Like you may have to like do a bunch of things to it to get it to work.
Right.
Which inherently
there's
just
like
memory wise is going to be a problem. Yeah.
right. And there is literally like spots in the ScanPy documentation that remark like the performance of this will be like somewhat different if you do it on sparse versus dense matrices. Which is, I don't know why, but for some reason there's some things that just matters.